Assalamu alaikum. My name is Entesar. I want to present my data in the title of Minocycline ameliorates hippocampal neuronal damage and beta-amyloid peptide deposition in LPS-induced Alzheimer disease rat model. This is the guidelines of the study. Alzheimer's disease is a neurodegenerative disease that is known to induce cognitive impairment in humans and animals. It affects approximately 7 million people worldwide and expected to increase to 12 million in 2050 unless significant breakthrough in treatment will develop. The cardinal feature of Alzheimer's disease is the accumulation of beta-amyloid peptide and the formation of neurofibrillary tangles that induce inflammation and neuronal damage. The underlying mechanism of Alzheimer's disease is still unknown, therefore no available treatment that really cure Alzheimer's disease. The only available drugs that has been approved by FDA just alleviate the symptoms but didn't treat the Alzheimer's disease. For rationale of this study, beta-amyloid deposit and neurofibrillary tangle formation are one of the hallmark and the underlying pathogenesis of Alzheimer's disease. And the development of therapies that targets amyloid are ongoing. So, the role of beta-amyloid accumulation and neurofibrillary tangle formation in pathogenesis of LPS-induced Alzheimer's disease rats model and the effects of minocycline on it are still not clear. So the study was conducted with the objective of to determine the effects of LPS injection on hippocampal morphology and beta-amyloid peptide deposition, and to determine the effects of minocycline treatment on hippocampal morphology and beta-amyloid deposition in LPS-induced Alzheimer's disease rats model in comparison to clinically approved and methyl D-aspartate receptor antagonist called memantine. This is the methodology. The rats were divided into 5 groups: control, LPS. The other 3 groups are LPS with treatment. The treatment was given from day 1 until day 4. And the treatment were minocycline 25 mg per kg, minocycline 15 mg per kg, and memantine 10 mg per kg. LPS was injected on day 5 at the dose of 5 mg per kg intraperitoneal. Then the treatment continued from day 6 until day 14. On day 28, the rats were sacrificed and the hippocampal were collected for further crescent violet and congruent staining. The statistical analysis was done using SBS version 24. This is the experimental timeline. For results and discussion, first, congruent staining, we have shown that in a CA1 region, there is increased number of amyloid, increase the amounts of amyloid in LPS group that reduce with treatment. Similar finding in CA2 region, there was increase the number of amyloid in CA2 region that decrease with treatment. Also in CA3 region, there is increased number of amyloid in CA3 that reduce with treatment. Similar finding in DG3 region, increase the amounts of amyloid deposition in DG region that reduce with treatment. For crescent violet, we do quantitative and qualitative. As shown here, LPS reduce number of alive cell in CA1 region, as shown here, and minocycline increase number of alive cell in CA1 region. For CA2 region, similar finding, LPS reduce number of alive cell as shown here, and memantine at both concentration increase number of alive cell in CA2 region. For CA3 region, same, LPS reduce number of alive cell and minocycline treatment at both dose increase number of alive cell. In DG region, same, LPS treatment reduce number of alive cell. As seen here, there is number of necrotic and pyknotic neuron. And minocycline treatment at different dose increase number of alive cell. Interestingly, the neuroprotective effects of minocycline in all region is dose dependence, means minocycline 50 got generate better neuroprotective effect than minocycline 25. LPS injection induced neuronal damage and beta-amyloid peptide deposition and neurofibrillary tangle formation in hippocampus, possibly due to inflammatory, oxidative, and amyloid pathway stimulation. This finding coincides with previous studies that have found that neuronal damage and amyloidosis in Alzheimer's disease rats model. Minocycline treatment reduced the neuronal damage and beta-amyloid peptide accumulation, possibly due to its anti-inflammatory, antioxidant, and anti-amyloid properties. The neuroprotective effects of minocycline was dose-dependent. This finding coincides with previous study that showed that minocycline can be used to treat a neurodegenerative disease due to its anti-inflammatory properties. Summary: LPS injection at a dose of 5 mg per kg once induces neuronal damage and beta-amyloid peptide deposition in hippocampus, and minocycline treatment daily for 2 weeks ameliorate these effects. So in conclusion, minocycline treatment attenuates the neurological deficit caused by LPS injection and can be used as preventive therapeutic agent to treat Alzheimer's disease.